Okay, so the topic that we are going to see are about biology, sub-branches and fields that has biology, and how did life start on Earth.
So, for understanding the topic, we need to know what is biology. Biology is the science of life. Its name is derived from the Greek words bios, that means life, and logos, that means study. Biologists study the structure, function, growth, origin, evolution, and distribution of living organisms. Now that we know what is biology, we can know about the branches that has biology. So here are some branches that has biology, but they are not all of them. The ones that are going to be mentioned are the ones that are the most important. So first we have biochemistry, that is the study of the material substances that make up living things. Then we have botany, the study of plants, including agriculture. We have cellular biology, that is the study of the basic cellular units of living things. We have ecology, the study of how organisms interact with their environment. Evolutionary biology, the study of the origins and changes in the diversity of life over time. Genetics, the study of heredity. Molecular biology is the study of biological molecules. Physiology is the study of the functions of organisms and their parts. Paleontology is the study of fossils. And finally, zoology, that is the study of animals and their behaviors. So, for the next topic we have, how did life begin on Earth? Okay, so some might assume that a scientifically minded person would pick evolution, and that a religious person will pick creation, but not always. The fact is, many educated people, including a number of scientists, question the validity of the theory of evolution. Scientists speculate about the existence of molecules that precede genetic material as we know it today. These molecules are supposedly more likely to arise spontaneously from inert material and are self-replicating. But science has found no evidence that such molecules ever existed, nor have scientists. They haven't been able to create any such molecule in a laboratory. Living things are unique in the way they store and process information. Cells convey, interpret, and carry out instructions contained within their genetic code. So, the beginning of life is still a mystery for scientists.
Hi, my name is Renata, and I'm going to tell you some characteristics of the living things and the difference between an eukaryotic and a prokaryotic cell. The characteristics of a living thing are that they all have generic code that follow the characteristics of the being, but in a microscopic level. They have homeostasis, which helps them regulate the temperatures of their body depending on their surroundings. They use reproduction to keep alive the species throughout the birth of different generations. They adapt and evolve depending on the circumstances and are what they need in order to survive. And they old and mature as time goes by. Now for the difference between an eukaryotic and a prokaryotic cell. An eukaryotic cell there are two types, an animal and a vegetable. The most notable difference among them is that the presence of the nucleus, the membranous compartment in which the genetic material is confined. A prokaryotic cell, they don't have a nucleus. The most noticeable difference is that the genetic material is found in the cytoplasm, in an area called the nucleoid. They are also unicellular and can be reproduced asexually or parasexually. They can also have different forms like coconuts, bacilli, and spiral shape. The components in a prokaryotic cell include plasma membrane, cell wall, cytoplasm, nucleoid, ribosome, prokaryotic compartments, and according to the type of prokaryote being analyzed, the cell may have Evangelia.
During the 1st century, glass had been invented, and the Romans were looking through it and testing it. They discovered that if you look at an object through the glass, the object will appear larger. These lenses were not used as much until the 13th century, when spectacle makers made those lenses in order to make glasses. In the year 1590, Zacharias Janssen and his father started experimenting with lenses. They invented the compound microscope, which is a microscope that uses 2 or more lenses in it. Galileo added a focusing device on the microscopes. Anthony Leeuwenhoek became very interested in lenses while he was working with magnifying glasses. He achieved lenses that were rounder and produced a greater magnifications. His microscopes were able to magnify up to 270 times. Robert Hooke, an Englishman, spent his life working with microscopes and improved their design and capabilities. Now I'm going to talk about the cell and the cell theory. First of all, what is a cell? Cells are the smallest unit of living matter. There are 2 primary kinds of cells. There are eukaryotic and prokaryotic. The cell theory is one of the basic principles of biology. It was made by the German scientists Theodor Schwann, Matthias Schilling, and Rudolf Virchow. The cell theory states all living organisms are composed of cells. The cell is the basic unit of life, and cell arises from a pre-existing cell.
Everything that enters or leaves a cell has to pass through a cell membrane, which is an organelle that is present in whole cell types. We have different classifications of this process in this transport. The first one is the active one, and this transport spends energy and goes against the gradients of concentration. We have also the passive one, that in this transport doesn't waste energy and goes in favor of concentration. It can be simple diffusion, vacillated diffusion, and ultrafiltration or dialysis. The simple diffusion is when the molecules cross the lipid bilayer, such as the oxygen and carbon dioxide that are gases. In the vacillated diffusion, the passage of molecules depends on the presence of the integral membrane proteins that allows this passage. Ultrafiltration and dialysis is when water or some solutes pass through a membrane due to hydrostatic pressure. We have 2 more classifications of cell transport, that is endocytosis and exocytosis. The endocytosis is the incorporation of materials into the cell, and the exocytosis is the secretion of the material produced by cells. We have the levels of organization. First one, we have the atom, then the molecule, next the cellular organelles, next the cell, then the tissue, the organ, next the organ system, the organism, the population, the community, then the ecosystem, and finally the biome.
We're also gonna be talking about homeostasis, organ systems, health and disease, and health in adolescents. But first of all, what is homeostasis? Homeostasis is the ability of organisms to preserve their conditions even if they are external or internal. Also, this has to do a lot with glucose, glucagon, and positive and negative feedback. Those are the main functions and the main parts that regulate the preservation of the body. So the main feedback that controls all of the functions of our body is the negative, while the positive is the one that appears when the negative feedback is decreased or even in some cases increased. There's also something called organ systems, which are a set of or a group of organs that operates a concrete function of our body. But for this we must be healthy, because health is not only the physical state of being in a good way, but also the mental and physiological state of being good. So whenever a disease comes, disease is a part that fights against this healthy lifestyle, and whenever this happens, it's because there is something in our organisms that it's like not working at all, and we must take care of it and have a healthy lifestyle. But there is also something called health in adolescence. Health in adolescence, it's very, very important, because in this part of our life we start growing up, and the maturity and emotional necessities starts to develop in our systems, in our bodies, and so we must take a lot of care of it.